Okay, Competency 10, Digestion and Absorption. You're going to find that information in OpenStax, Chapter 23, and Patent Chapters 40 and 41. Again, there's a link. The next slide has more links, but you can access this slideshow in through Blackboard under the Module 4 Videos and PowerPoints tab. So we're home stretch, Competency 10 of 12. So a couple of things we want to go through. Again, I'm going to keep this video brief. Before I go into that, this link, it says digestive review questions. I wanted to add to that. I tweaked 10, 11, and 12 review question format. You'll see that, I'll click on it now. You'll see that format is a little bit different than what you have had. I have the complete questions and options. And then at the bottom, when you scroll down, a lot of questions. These are from your test bank. So again, these are the exact questions that you'll see in your test from the test. This is the test bank. That's what I'm trying to say. So you're going to have random 40 of these 147 questions. But you'll notice the multiple choice number, and then it says reference page 903. That would be helpful if you have the patent text. So you don't need it, but if you do have it, that's what the page number references. More importantly is the topic that it discusses. So anyway, as you review or before you take the exam, you may want to go through some of these. They match up with the PowerPoint. They match up with the chapter. So anyway, let's continue with this. I want to get that back to full screen. So anyhow, that's what you'll find. 11 and 12 are the same way. The links for these are in the same place. You've seen them in previous modules under the practice tab for each of the units. So anyhow, let me get the captions back up. There we go. Okay, so again, I'm going to keep this brief. There are recommended videos for you. These go with the Padlets. The Padlets, if you haven't noticed already, the Padlets are set up to go with the competencies and to go with the learning objectives under each competency. So the state of Wisconsin, as you know, or as a reminder, sets up through WIDS, W-I-D-S, and through the Wisconsin Technical College system. You have access to that document through Blackboard, and it shows you the learning objectives and the competencies. So there are 12 competencies for this course and anywhere between 4 and 8 learning objectives under each competency. So these Padlet videos, as well as our discussions, the PhysioX activities, and the case studies all relate to those objectives under each competency. So some of the items in our discussions in the slide shows do have there are some slides I've left in there, some slides that I've put in there that maybe don't directly line up with an objective, so I may not spend much time on it or even any time on it. It may be more or less FYI type of stuff. So anyway, keep that in mind as you go. But these recommended videos do line up with the learning objectives. So if and there are obviously others, there are thousands of videos in YouTube. You have access to what the learning objectives are in Blackboard. In fact, I put those learning objectives within each competency and within each unit under the modules. So anyway, you can YouTube search any of those objectives if you're struggling, and you'll come up with a lot of other videos beyond what I have here. So without further ado, let's get going. Digestive anatomy you had in general. So again, mechanical and chemical digestion both start in the mouth. We chew the food, we start breaking down some sugars, we go through deglutition or swallowing, and then a series of rhythmic motions called peristalsis that are going through the smooth muscle tube or tract that takes us down into the stomach. So the upper digestive tract from mouth to the stomach. Okay. And then once we're in the stomach, we're going to see the continuation of chemical and mechanical digestion. We do start some protein digestion as well in the stomach. So we'll look at a few of the hormones and a few of the chemicals involved with the stomach. We'll look at a few with the salivary glands as well, the accessory glands. And then as we get into the lower GI tract, we pass through the pyloric sphincter into the duodenum, the first small, short section, maybe a foot or so, 15 inches at the most of duodenum. And that's going to head into the jejunum and the ileum of the small intestines, upwards of 20 feet of tubing there. And then this is where we're going to see more hormones, more chemicals, and a lot of activity regarding digestion, assimilation, absorption, secretion as well. Going back up let's just continue through the tube. So anyway, we leave the small intestine, we enter into the large intestine. We see the ascending colon or the cecum first, and then the appendix off of the cecum, and then the ascending colon, transverse, descending sigmoid, and then into the rectum. So going back up and looking, pardon me, briefly at the accessory organs of digestion, we see the liver, gallbladder, and pancreas being important features, and they're all going to have ducts that lead together and dump their fluids, primarily digestive enzymes, into this area where the stomach opens up into the duodenum or the small intestine. So we'll spend some time looking at some of the chemicals of digestion that are going to come from these organs. We've looked at enzymes up to now. So anyway, a lot of this information isn't going to be that terribly new. So again, I don't want to spend a heck of a lot of time on it. You've been doing a great job, all of you, critically thinking, problem solving, digging deeper, taking charge of the learning process this semester. So I really appreciate that. And every semester that you have an online class from here on out, they may be more frequent. Keep doing what you're doing. And if your instructor, if you have suggestions for any of us, please let us know. We'll add and subtract and do what we can to enable you to have the best experience, because it is more difficult. You're realizing it, maybe, but I think it's more fulfilling to do more of to take part in the learning process more. So anyway, I don't want to spend much time on this. I think that all of you are doing well, and will continue to. So look through these. Don't forget there are notes within the PowerPoints. When you look at these and open these up, you will see notes at the bottom of the slides. So make sure you look at that. But the primary aspects of digestion, I'd mentioned earlier, we're going to see secretion of enzymes, absorption of nutrients, elimination and excretion of waste, and ultimately there's going to be some hormones involved. Whenever you see regulation, we're going to see a neuroendocrine aspect. So regulation always means hormones. So we will see hormones as well as enzymes. Motility has to do with the ability to move something, or for that structure to be able to move. So anyway, mechanical digestion, we saw mastication. This really occurs throughout the digestive tract. It starts in the mouth. We see this continuing in the stomach. We break particles down with our smooth muscle of the digestive tract. Deglutition is that first stage where we, after chewing and mastication, the first step toward removing the food from the mouth and getting it to the stomach is via what we call deglutition. It's a fancy word for swallowing. So anyway, oral stage, pharyngeal stage, esophageal stage. Okay, we see the chewing. We're going to use the tongue and the molars and form what we call a tiny bolus. And we're going to have multiple boli or boluses that we're going to swallow. We see multiple muscles involved. We get into the pharyngeal stage, and that epiglottis is going to close as the tongue presses down and pushes that bolus backwards. We're going to see that epiglottis close the respiratory tract off from the digestive tract, and then we can continue into that esophageal stage of deglutition and peristalsis. So part of peristalsis is kind of just going around and squeezing the bolus further down. But another thing we can do, so you can see the contraction behind the bolus to move it forward, that's peristalsis. Segmentation means we create segments and then kind of trap the bolus in a spot, and then we exert some force in between and get that the large globule to break up into smaller globules. So the bolus becomes a bolette, I guess. Okay, so again, motility having to do with movement, churning in the stomach, propulsion, retropulsion, kind of like a blender activity. We're going to see as we leave the stomach, we're going to form kind of a soupy mixture called chyme. And that chyme gets ejected about every 20 seconds into the duodenum, and when something's in the stomach, of course, and that takes anywhere from 2 to 6 hours to empty the contents of the stomach. So because protein digestion is occurring in the stomach, we typically see heavier protein meals staying in the stomach longer, whereas meals that have more carbohydrate in them are going to be in there a little shorter. So carbohydrates in the stomach, less time, proteins and lipids, maybe a little bit longer. So, and that 6-hour mark, it could be even more than that. So anyway, gastric emptying is controlled certainly by hormone, the neuroendocrine connection. Everything is controlled by the neuroendocrine connection, right? So GIP, gastric inhibitory peptide, fats, once we get into the duodenum, fats are getting in there. We'll see the stomach's churning start to slow down a little bit. We see insulin production coming from the pancreas. So basically what this is saying is once action starts to move away from the stomach and into the duodenum, where again, we're going to see pancreatic and liver and gallbladder enzymes and emulsifiers being released into that duodenum area. That's going to trigger GIP to be released, and that's going to kind of slow the stomach down and basically continue to keep the train moving. Get out of the duodenum, keep going, and get the stomach to slow down a little bit. So anyway, peristalsis is going to be regulated by cholecystokinin. This is a long one. That's why we do CCK. We've seen this before. Kinin, of course, means movement. Cholecyst has to do with the gallbladder. So we'll see cholecystokinin playing a role throughout. It's kind of a common theme during this digestive chapter, or this digestive competency. Yeah. So peristalsis rate picks up as the chyme approaches the end of the jejunum. So we leave the stomach, we get into the duodenum. Things slow down in the stomach. We're starting to speed things up a little bit, get out of the duodenum, which is, again, a fairly short trip. And then we're going to be in the jejunum for a brief period of time. It's a long trip, though. We're talking maybe 8 to 12 feet in the jejunum. So anyway, we're going to keep the train rolling. Peristalsis. So once we're in the duodenum, CCK, again, triggered by, this is just another domino that falls along with GIP from fats. We're also going to concurrently get CCK released, and that's going to trigger the smooth muscle of the entire small intestine to squeeze and go through peristalsis a little bit quicker, or a little more rapidly, to get things moving toward the large intestine. So anyway, chyme leaves the stomach. We enter, so 2 to 6 hours in the stomach, and then only about 5 hours. And again, in the small intestine, the stomach is only, maybe from entry to the stomach to exit of the stomach, you're talking, maybe a foot and a half at the most. And the small intestines, we're talking probably close to 20 feet at least. 5 hours, so we can go 20 feet in 5 hours through the small intestines. It takes us 2 to 6 hours, so roughly the same amount of time to go 1 foot in the stomach. And that's because the stomach is doing a lot of work forming this chyme. So chyme is really a blended soup that makes life easier when it comes to assimilation and digestion. So if you haven't done the PhysioX for digestion yet, please do. A lot of this will make more sense to you once you do that. If you've already done the PhysioX for digestion, a lot of this makes sense. So in Wet Lab, we do a really good couple of activities that are similar to the PhysioX. We also look at surface area. You can get faster absorption with when you decrease surface area. So when we take a big chunk of something and break it into a million little tiny chunks, we speed up the rate of digestion and absorption and assimilation. So that's why we spend so much time in the stomach, is because once we're ready to go into the small intestines, we need to be ready to absorb and assimilate nutrients into the bloodstream. So anyway, hydrolysis, that just simply means, again, we add water on the front end. So hydration is important. There's a lot of hydration within the digestive system, certainly available. There's a lot of water available. So adding water to something will break it down, will catabolize it or metabolize it. So chemical changes result from adding water. is what this means. And then we add enzymes along with water. So enzymes will speed up the reaction, speed up the metabolism, the catalysm that's going on. So catabolism, okay, right? Catabolism, catalyst, big becomes small chunks, okay? Add water, add enzyme, boom. Remember, enzymes are protein-based too. So you want to remember that, even though enzymes do work on all of the macromolecules. Enzymes are very specific to temperature and pH, especially. So one of the wet lab activities we do when we're face-to-face is expose enzymes of proteases, lipases, specifically some starches as well. We expose those to enzymes, and so we have amylases going after starches, proteases going after proteins, lipases going after lipids. So what we'll do is heat up the temperature of the environment to see if the enzyme works as well. We'll cool the environment and see if the enzyme works as well. We'll add acid to the environment to see if the enzyme works as well. We'll add base to the environment to see if it works as well. So anyway, we can alter the conditions of pH and temperature and then monitor how well the enzyme works. And we've done this for several years and found that, in fact, temperature and pH do affect enzymes. They don't work as well if they're outside of fairly small ranges. So anyway, that's why a fever inside the body can affect enzymatic activity. A long-term febrile condition can lead to some major issues, certainly systemic-wise. So anyway, enzymes, cellular enzymes too, not just digestive enzymes, but enzymes, period, are very specific to temperature and pH conditions. So continually being destroyed and eliminated from the body, certainly. So we have what are called proenzymes, or kind of like pre-enzymes. Usually we'll see the suffix gen. Gen means it generates whatever is in front of it in the word. So trypsinogen generates trypsin. Chymotrypsinogen generates or is the precursor to chymotrypsin. So anyway, that's what all that means. So carbs are first. We'll zip through these. Carbs are saccharides. So whenever we see saccharide, we know we're talking about sugars. We think of polysaccharides as long chain carbohydrates. We certainly, again, hydrolysis and water in the form of water and amylase. These are the different types of amylases are going, again, we see ACE, ASE, and we know we're dealing with an enzyme that breaks down amyls or saccharides. So long chain saccharides get broken up into disaccharides by specific types of amylases. And then we can continue that process with, so the main disaccharides are sucrose, lactose, and maltose. Starch would be the polysaccharide we're most familiar with. So these particular enzymes, sucrase, lactase, maltase, are going to be added to the disaccharide that goes with it. So sucrose is the disaccharide. We add sucrase, and it breaks it into a monosaccharide, and so on. So actually 2 monosaccharides. So we'll see glucose, fructose, and galactose are the 3 main monosaccharides. So anyway, polysaccharide, we see long chain carb. The mouth is where carbohydrate digestion begins. So we're going to release salivary amylase, and we're going to get hydrolysis to occur. We're also going to see carbohydrate digestion continue in the duodenum of the first section of small intestine, where we're going to release pancreatic amylase as well. And we're going to see all of these long chains become a bunch of disaccharides or two-chain carbs, and those would be in the form of sucrose, maltose, or lactose. And then we keep the train rolling, and then we add these, what are in the small intestines, we have what are called brush border enzymes, sucrase, lactase, maltase, that are going to break up the specifics into either glucose, fructose, or galactose. And then protein digestion primarily is going to begin in the stomach. We see protein being long chains of amino acids. So we're going to see what are called proteases and water that are going to start breaking intermediate compounds of proteins into amino acids. Okay. The main proteases we're going to see are pepsin in the stomach. Hey, pepsin, that's your first protein digestive enzyme, protease, called pepsin. And then we continue, we get what's called trypsin coming from the pancreas. So remember, the pancreas is secreting digestive enzymes as being part of its exocrine umbrella. Remember, the pancreas is exocrine and endocrine, so we're primarily seeing it as an exocrine gland in the digestive system. So anyhow, trypsin in the pancreas being released into the duodenum. So pepsin starts the process, trypsin continues it, and then those brush border enzymes for proteins are going to be called peptidases. So we see this term peptide in there. And again, peptide just simply has to do with a single protein. Amino acids form peptides or polypeptides, ultimately becoming long chain proteins. Gastrin is one of the hormones in the stomach, along with CCK from the duodenum, and that's going to trigger some of these proenzymes from the pancreas, primarily trypsinogen and chymotrypsinogen. Trypsinogen, remember, is going to become or lead to or activate trypsin. So trypsinogen is the precursor, the activator for trypsin. And we saw just in the previous slide that in the duodenum is where we're going to find trypsin secreted or released from the pancreas. So basically, once food gets into the stomach, once the boluses start getting in the stomach and start getting churned up or mechanically digested, gastrin gets released from the stomach into the bloodstream. CCK from the duodenum into the bloodstream. That's going to trigger the pancreatic proenzymes or preenzymes, trypsin and trypsinogen and chymotrypsinogen, to become activated, which then leads to those guys activating what we're ultimately going to need, trypsin from the into the duodenum and chymotrypsin from the pancreas. And what's going to happen is trypsin attaches to chymotrypsinogen and activates chymotrypsin. Okay. These are their main protein digestive enzymes. Okay. So we look at it in a little more detail in a graphic. We see long chain proteins in the stomach, proteases. So we see pepsin. Okay. This is our first one. Okay. And then from there, we're going to head down into the duodenum, and we're going to see intestinal peptidases. We're going to see proteases from pancreatic juices that we just saw, trypsin and chymotrypsin, ultimately breaking up these long chain proteins into amino acids. And then these amino acids are going to be absorbed into the bloodstream and delivered to our cells for protein synthesis. Fat digestion, we're going to see the beginnings of fat digestion in the duodenum or the small intestine. Okay. This is where we're going to see this emulsification process performed by the presence of bile. And bile has what we call bile salts as well as lecithin in it. And what we're going to get are these little micelles. So we have oil and water, so the oil being the lipids or the fats, and we're going to see this lecithin from the bile is going to attach to this oil and form basically what we see as a mycele or a shell. So we get this hydrophilic shell. Okay, so the shell has an affinity for water. The globule does not, right? The oil globule is hydrophobic. So what we have with these micelles, with this lecithin portion of bile and these bile salts, is we're going to be able to coat those fat globules, and we're going to break them down into smaller bits. So these large fat drops or globules they're too big to be absorbed in the small intestinal wall, so that we form a mycele, and then from that mycele we can then add pancreatic lipase, and the pancreatic lipase can come in as the mycele breaks apart. We're going to see smaller portions of that original triglyceride to become monoglycerides. Okay? And those monoglycerides and fatty acids are going to get, lipase is going to help the process of breaking those myceles up. So we go from triglycerides to fatty acids, glycerol, and monoglycerides. So there's an intermediate process involving bile. And again, bile is going to be comprised primarily of lecithin and salt. So we're going to coat these triglycerides. Then we can add a little lipase, and that lipase is going to come in and start breaking up these chunks of fats into smaller, more absorbable bits. So we see secretions throughout. We've already mentioned the main ones. We have saliva and salivary amylase. We've got pepsin in the stomach. We've got hydrochloric acid as well in the stomach, secreted by these parietal cells. And again, pepsin is an enzyme, and pepsin starts breaking down these big proteins, and it only works in an acidic environment. So that's why hydrochloric acid is there, is to enable pepsin to function properly. Okay. So that gastric juice is primarily going to be pepsin and hydrochloric acid. We're going to see something else called intrinsic factor released by these parietal cells, and that's going to relate primarily to vitamin B12. So it's kind of like a key to help with B12 absorption in the small intestines. So folks who are lacking intrinsic factor from the parietal cells of the gastric wall are going to maybe have problems with the absorption of vitamin B12, and that could lead to a whole plethora of issues, particularly with memory, with muscle coordination. Anyway, so they've done research with dementia and Alzheimer's and Parkinson's regarding B12, intrinsic factor, and parietal cells of the gastric mucosal lining. So anyway, there's a couple other, we of course have mucus, we've got water, we have what are called chief cells and parietal cells, again, secreting that intrinsic factor. Chief cells are going to be what are secreting the digestive enzymes. Okay, we see hydrochloric acid also secreted by parietal cells. So HCl, an intrinsic factor, secreted by parietal cells. Pepsin, primarily secreted by those chief cells. So as we travel out of the stomach and get that chyme going into the first spot or first stop of the small intestines, the duodenum, we can see where that cystic duct and the hepatic and bile ducts form and come together to form the common bile duct. Okay, so cystic, hepatic, common bile duct. Okay, so that common bile duct continues to travel to an opening, a little ampulla there where the pancreatic duct connects with it. Okay, so we get a lot of the pancreatic digestive enzymes, all of these liver enzymes and gallbladder particularly bile being secreted into this spot. So that's why I put this in here just again as a visual reminder of where we're heading. When we look at the pancreas, we see the acinar cells primarily secreting these proteases. We do have lipases and amylases as well as nucleases coming from these acinar and duct cells. Again, proteases go after proteins, lipases go after fats, and nucleases going after DNA and RNA that's found in the foods we consume. And then certainly amylases, we know, go after starches or sugars. Bile, we looked at a little bit ago. Again, lecithin and bile salts primarily. We associate those as forming little spheres or micelles. These little micelles, again, are going to assist with the breakup of large fat globules once we add a little bit of lipase to the mix. Okay, and then we have some sodium bicarbonate also coming from the within bile and coming from the liver. Okay, so remember too, bile it's secreted by the liver. Let me back up to this. So bile is produced and secreted by the liver. So we talked about it in general, but students still sometimes trip up on that one. So you want to remember bile is produced and secreted by the liver. It's stored in the gallbladder and concentrated in the gallbladder. So bile is really powerful when it's produced. So when we put it in the gallbladder, it gets a little bit more concentrated. So then we don't have to use. Anyway, bile's powerful. So we can get it even more concentrated. So then we just have to do little spritzes of it to get a lot of action occurring. Okay, so it's concentrated in the gallbladder, stored and concentrated, secreted, produced and secreted by the liver. We've got sodium bicarbonate that also comes from the pancreas, and then certainly cholesterol, which are products of the detoxification of toxins in the body. And then we also have what are called bile pigments or colors, such as bilirubin. A lot of this ends up in the fecal matter, by the way. Some folks have what's called a fatty stool. So they poop and the stool has almost like globs of fat floating in the tank, or, not in the tank, in the bowl where you poop. Hopefully you're not pooping in the tank. Upper decker, right? You want a regular decker, but fatty stool means we may have a problem with bile. We may not have enough bile. We may have a blockage. Sometimes gallstones could get stuck in that duct, the cystic duct. So we're not getting enough bile released. So we end up with some of the fat not getting absorbed in the body, and it ends up in the stool. So, but anyway. Intestinal juice. So anyway, we're now in the small intestine. Again, we're going to see some of these brush border enzymes, but we see mucus and water, sodium bicarbonate, of course, salivary secretion. When do we start So we go from small intestine, put on the brakes, and go back up into the mouth. And so the olfactory, this basically is talking about the nervous system, the control of secretion. So olfactory will start stimulating saliva. Visual, we call this the cephalic phase of digestion. So that can trigger the stomach as well. To start, we start getting the vagus nerve kind of jolting the stomach a little bit. We get gastrin release, and remember, gastrin is what's going to tell the pancreas and the duodenum to get ready, to get trypsinogen and chymotrypsinogen activated. So gastrin is that a major domino to start falling. So that smell phase, sight phase, hearing, pots and pans clanking, whatever it might be, the sound of other people chewing. I mean, I don't know, maybe that shuts off digestion for some people, right? Some people can't eat in public. So anyway, they call that the cephalic or psychic stage or phase. It's a lot of mental factors. And then the gastric phase occurs once we've entered the stomach, and the stomach starts doing its thing. Gastrin again is continuing to, so gastrin's getting, so the cephalic phase will get gastrins triggered. And then once we get into the stomach, gastrin's ready to roll. So it gets released. And then that's going to trigger, again, then the intestinal phase after the gastric phase. And pancreatic secretion, again, primarily dealing with CCK or cholecystokinin, as well as one called secretin, produces pancreatic fluid that's low in enzyme content but high in bicarbonate. So secretin is going to get triggered when we have a maybe a pH issue with the stomach. So as contents leave the stomach and enter the duodenum or small intestine, the pH of that chyme is like a 2 because of the hydrochloric acid. And so secretin will come in and trigger bicarbonate explosion, so we can start getting that pH back closer to neutral. So anyway, that's what's going on there, because we do want to see a decrease in hydrogen ion concentration, or an increase in pH, so up to close to 7 or even 8. Okay. Increase in excrete. So CCK triggers the pancreas to release its digestive enzymes, its proteases, amylases, and lipases. Inhibits hydrochloric acid secretion. So we want to slow down the amount of acidity that's coming in to the duodenum. So again, pH balance is controlled by secretin and CCK. And then we also need the gallbladder to start spritzing bile. So liver continually produces it and spritzes it. Again, secretin and CCK are going to trigger the release of bile from the liver and gallbladder. Absorption, this is simply means getting nutrients absorbed into the bloodstream. So we primarily are going to see this happening in the intestinal mucosa. Okay, so absorption occurs in the small intestine primarily and goes into the blood or lymph systems, one or the other. And of course, we know they're interconnected. So we're going to see if we're putting something into the blood, right away your brain should go to capillary networks, right? So we've got a lot of capillary networks in the small intestines. And we do have secondary active transport. We're trying to push sodium against its concentration gradient. So we need active transport. We have co-transport. This is how glucose goes. Glucose is going to piggyback or get co-transported along with sodium. So here we can see what we call a lacteal. That's a fancy word for a lymphatic vessel that's surrounded by capillary networks. Yeah, the presence of these helps to clean. It helps with lipids. It helps also with some of the cleaning of whatever it is we might have consumed. We see whenever we're putting something into the bloodstream at a capillary network, such as in the lungs or in the small intestines, we're going to see a higher presence of lymphatic tissue, whether it's lymph nodes or lacteals. So we do see a lot of lymph nodes in the lungs and the capillary networks around the alveoli and around the lungs. We also see these lacteals and lymph nodes as well around the small intestine. Okay. Absorption, again, we're going to get absorption into the bloodstream, travel right to that portal system, that venous blood supply that's heading from the lower body and the basically anywhere below the liver. All the blood that's coming back to the heart is going to stop and pass through the liver, and that includes this portal system from the small intestines. And then elimination, of course, getting rid of the fecal matter. Defecation is the fancy word for that. Constipation, holding on to too much, it could be a sign of dehydration. So your body is trying to retain water as much as it can. Diarrhea, possibly due to some sort of bacterial infection. Certain foods, food poisoning can certainly cause that. So, anyway, that's pretty much it. Nutrition and metabolism. Most of this we've gone through extensively throughout. There are a couple of charts that you can certainly go through and look at. ChooseMyPlate.gov if you feel like looking into that. Canada has its own Eat Well plate, it's called, so you can take a peek at the differences in those. Again, catabolism, anabolism, right? We're breaking things down and we get energy from it, chemical energy, in addition to getting these micronutrients. Anabolism is building up or synthesizing. So this takes place inside of cells primarily. Okay, we've talked about ATP. So that's the carbohydrates we associate typically with that. Polysaccharides, disaccharides, monosaccharides. So go through and list some specific examples of polysaccharides here. And then the 3 disaccharides, and then monosaccharides, glucose, fructose, and galactose. Then, of course, glycolysis occurring in the cytoplasm. We've looked at this as well earlier in the semester and last semester a little bit. Cellular respiration continues in the citric acid cycle. That takes place within the mitochondrion. Okay, and then the electric transport chain also takes place in the mitochondrion. So from there, we're going to generate roughly 36 molecules of ATP. And we're going to be left with some H2O, some water. We're going to be left with carbon dioxide when it's all said and done. And if we don't include the citric acid or Krebs cycle, that means oxygen is not involved. So the anaerobic pathway simply involves glycolysis, which is occurring in the cytoplasm of the cell. And so your glucose goes through glycolysis out here. That's the anaerobic pathway. And then you can see CO2 as a byproduct. We get pyruvate, and those pyruvates are going to enter into this mitochondria if we have oxygen present. If we have oxygen present, we can continue the process of into the citric acid cycle. We have a coenzyme that helps to break everything down as well, and we get the carbon dioxide released from the citric acid cycle. And then as we get all of these hydrogen ions, we can continue the process of ATP gathering when we, as well as water release in this electron transport system. Okay, so citric acid cycle, electron transport chain, traveling along, getting all kinds of ATP, releasing water, releasing carbon dioxide. And again, when we just do glycolysis, our net gain of ATP may only be 2. Okay. As we go down the line through the citric acid cycle and the electron transport system will give us 36 ATP. And glycogenesis, we're going to generate glycogen. Okay, glycogen. Glycogen is stored glucose. Okay, and then when we need to break up the stored glucose, we call it glycogenolysis. So when we are lacking glucose, glucagon is going to come in and trigger glycogenolysis to start breaking up stored glycogen into glucose bits. And then insulin comes in and grabs those up and takes those to the cells throughout the bloodstream. Cori cycle, that's the lactic acid production that occurs during glycolysis, which again is anaerobic. So lactic acid, and where does that lactic acid end up? Where does lactic acid end up? It ends up in the liver. Okay, so we've got the liver, or we've got the muscle cell, and we're producing some lactic acid. We're going through glycogen, glucose, lack of oxygen. Lactic acid builds up. It's going to get taken to the liver. A little hepatocyte or liver cells are going to take that lactate, convert it into pyruvate, glucose, and stored glucose in the form of glycogen. And then we can deliver that glycogen to the muscle, and we can break that glycogen up into glucose, called glycogenolysis, and so on. So that cycle can continue recycling lactic acid. Hey, blood glucose levels, again, hyperglycemic. If I'm hyperglycemic, generally I want to be right around 100, right? So if I get much higher than that, I'm going to pee it out potentially. I may store it as fat. I may store it, go through what's called glycogenesis, where we're going to store glucose as glycogen. I may just use some of it up right away as energy. If I'm hypoglycemic, I don't have enough sugar, what I'm going to do is go through the stored glucose, this glycogenolysis is stored glycogen. I'm going to break up that stored glycogen, and that's going to give us some glucose. Or I'm going to create new glucose, gluconeogenesis. Okay. So we can do glycogenesis. okay, generating glycogen. Okay. And it's insulin gets blood sugars out. Glucagon is going to increase blood sugar levels by triggering some of these factors that we see over here. So when blood sugar is low, glucagon is going to trigger glycogenolysis, glycogenesis. Okay. Gluconeogenesis as well. We have stored. We can use fat. Okay? We have glycerol from fats that we can convert into glucose. We can take amino acids or proteins, bring them to the liver, and convert that into glucose. Okay, so gluconeogenesis coming from fats or proteins. Glycogenesis is stored glycogen being converted into glucose. And again, insulin is going to get triggered after, certainly after a meal, or if during fasting, in between meals, if we've got a glucose deficiency and glycogen gets converted into glucose, then we would see an increase in insulin that way. Certainly epinephrine will trigger glucose synthesis as well. Okay. So anyway, triglycerides, you can look through those. We talked about fats a little bit, and fat catabolism or fat breakdown. Okay, we're not going to get too detailed. We know that hydrolysis is involved, and we also know that lipases are involved. Okay, glycerol. And remember, we can use glycerol to take to the liver and go through that citric acid cycle. If you take a nutrition class, you would go through this in a lot more detail, but since this isn't a nutrition class and it's not part of the objectives, I'm not going to spend much time on this other than some of the very basics. Carbohydrate, lipid, and protein metabolism. Again, we've gone through carbs plenty. We've gone through lipids, the basics of lipids. Okay. Insulin, growth hormone, glucocorticoids are all involved in keeping our fats organized. And protein, we've gone through protein metabolism and protein synthesis off and on really throughout the semester. Okay, so protein mobilization and protein catabolism. Again, we can, through ribosomes and rough ER, we can convert proteins into amino acids and kind of keep them stored in the cells. Now, if we need to break down some of those proteins, we can go through some hydrolysis. We have some proteases inside the cells, so we can then get amino acids that way. And certainly the liver, through ketones and deamination of amino acids, getting rid of an amine group is all that means. That will form urea. And then again, as we break down amino acids, we're going to get ketones. This is out of that keto diet. People consume excess amounts of amino acids or proteins. Well, those can get broken down by the liver through gluconeogenesis, and we can get glucose that way. It's the third most preferred form of getting glucose, by the way. Okay, so number 1 is carbs, number 2 is fats, number 3 is protein. Okay. We do get some, but man, that should not be your primary source of glucose. You need 70 grams of glucose per hour, minimally, just to be able to function at just to be able to sleep, basically. So we're probably burning 100 to 200 grams, maybe an hour. So it's coming from storage units glycogenolysis from stored glucose. We can store about 70 grams in the liver of glucose. So if we need 120 in an hour, we're going to get 50 from muscle, likely. But if we need more than that, we can certainly get it from lipids. We can, through gluconeogenesis from fatty acids and lipids. And then lastly, we can get it from keto acids, glucose. So folks who are malnourished or not eating properly certainly will go through the process of breaking down proteins and amino acids from cells, and then that can lead to that. And then so I'm dumping glucose into the blood. So now I'm also dumping insulin into the bloodstream. So anyway, either way, insulin is desirable to get glucose transferred to the cells. Now, whether that glucose came from carbs, proteins, or fats, well, hopefully most of it's coming from carbohydrate. Anyway, that's that. A couple things with vitamins and minerals. You certainly are welcome to go through metabolic rate. This is highly debatable. BMR, base metabolic rate. Yeah. Anyhow, that's that. You can go through those on your own, and for more info. If you have questions, certainly feel free to email me and let me know. Have a great day.